My name is Clarissa Molina. I'm a third-year PhD student in the Department of Biological Sciences at the University of North Texas, and I study wild turkey. My research focuses on population and landscape genetics of wild turkey, so I'm trying to determine in one area of Texas how many populations we have and which ones are more at risk to extinction, to inbreeding, and which populations are doing well, and the distance, geographical distance those populations are to each other. And I want to essentially give these findings to agencies and landowners so that they can create management strategies that will benefit and promote wild turkey conservation in Texas. And then in East Texas, I'm primarily focusing on evaluating a recent restoration attempt for wild turkey. So I'm trying to figure out, it seems to be working, and I'm trying to figure out why it worked, and I'm using genetic data to do that. So figure out why it worked and how we can replicate that success in other areas. So to bring back the wild turkey to its native range, where over the last 100 years it has been not doing very well. Their populations have been very fragmented and isolated, and have been very vulnerable to disappearing.
Awesome. Oh my God. So what sparked you into you choosing the research you're doing now?
Yeah, so I was really interested and passionate about promoting and preserving global biodiversity. So that's very broad, right? And so really any project that allowed me to investigate certain questions that would accomplish that end goal, right? So that was what ultimately sparked my interest in this kind of research. Specifically, I had worked in the GIS department for UNT facilities, and I was really interested in applying the skills I'd learned making maps and tracking movement. So I'd done a research project studying movement of painted buntings in my undergraduate. So I was really interested in applying those same skills. And then I had no experience in genetic analysis, but I was really interested in it. I had taken genetics in my undergrad, and I had no idea you could blend those two genetic and spatial analysis techniques together into one project. So when I learned about the potential for the field of landscape genetics and the opportunity to study wild turkey, and the fact that wild turkey is a native species that is declining across the United States, and there's a lot of active restoration work, I wanted to be a part of that unified movement to restore wild turkey, to protect their populations, and to support them to ensure their future.
That is awesome to know. Wow.
Yeah.
So what does a typical day for you look like, especially when you're doing your research?
So a typical day changes. Some days I am hunched over my laptop, doing a lot of administrative tasks, answering emails, writing reports. Other days I am working all day in the lab. I'm pipetting, I'm extracting DNA, I'm conducting PCR, so I'm amplifying my DNA or sending it off, driving down to Fort Worth to send it off for sequencing. So my day really changes. Sometimes I work in an office in the environmental science building. Sometimes I work in the life science building. Sometimes I work from home. I work a lot with other people as well. So I have several great, fantastic technicians that we work together in a team. So a lot of my time is spent collaborating with them. And so it's a very people-oriented project. So I spend a lot of time with other scientists and with other graduate students, other undergraduate students. So a typical day just varies. Yeah.
That is awesome.
Yeah, every day is different, which is really a really exciting thing.
At least you're up on your feet, like at some point. Yeah.
Yes, yeah. And I like the change that, depending on what I'm doing and what phase I am in my project, what I'm doing right now looks entirely different from what I was doing 6 months ago.
Yeah.
Sometimes I'm out in the field collecting my data, chasing turkeys, and being outsmarted by them at every turn.
That
so
is
it
awesome.
just depends. Mm-hmm.
Every day's a new adventure, that's for sure.
Yeah. Yes, yeah, definitely.
So why is your research important?
So my research on its own, my research is really just a drop in the pond of the research that is being done to um, protect, our global biodiversity and to protect wildlife. So my research is really, I like to think of it as like a brick in the wall that we're all building. And so for me studying wild turkey, my research hopefully benefits not just the wild turkey, but everything that the wild turkey, all living things are connected. People talk about the web of life, or when one tugs on one thing in nature, he finds that it pulls on everything else, right? So protecting one part of our natural system will ensure that the other parts can continue to function, because everything relies on each other.
Yes.
And so in the broader sense, like, my research on protecting and restoring and wild turkey populations is also trying to preserve the landscapes and the wildlife that we have as a whole. But also, I think wild turkey are important because they're also Texans. Like, they live here, they're native to Texas, and so the fact that they're disappearing because of something that we are doing, because of the destruction that we're causing or our actions, makes me feel even more the duty to try and negate those threats that they're now facing because of us. So my research is important as a contribution in that effort to kind of reverse some of the damage that we've done, because humans have a lot of power, and we can use that for good, or we can use that for harm. And so I want to try to bring more. I want to try to use the power and the influence that we have to preserve and to protect and to be good stewards.
That is awesome.
Yeah, of course.
So, how do you feel about the current accomplishments and findings that you've had during your research?
So as far as the findings, we're still a little too early in the game to be able to tell conclusively what our findings are. But as far as our accomplishments, I feel very proud of our team. So I have, like I mentioned, a very dedicated team of undergraduates that we're all working together to bring about this research. So I'm very proud of the strides that we've been making in the process so far, and I'm really looking forward to what our results will say about the questions that we have, our research questions and our aims.
Oh, that is awesome. Okay. How is or will your research that you're doing now contribute to like a real world problem that we have now?
So I would say a real world problem that we're currently facing is an unprecedented loss of our wildlife and our landscapes that are precious. They're like not only to humans, because we do rely on the ecosystem services that the land and that the animals on the land provide for us, but also just inherently, I think that our land and our wildlife are inherently valuable. So the fact that we are losing a lot of it at an unprecedented rate is really discouraging and really scary. But my research, like I mentioned, I don't think I'm gonna single-handedly save the world or anything like that.
I mean, you could.
I don't know. I mean,
well, who knows? We'll have to find out at some point.
No, and that honestly, I think a lot of people like view scientists as like they're just like working alone in a lab, and like they're gonna like come out with eureka, and it's like all the solution to all the problems are gonna be in their hand. But I'm like, science is so much. It's like so much more about baby steps and filling in the knowledge gaps that we have. Like, the phrase "standing on the shoulders of giants." Like, my research, is buttressed and founded on people who will come before me, investigating into how these systems work, how we can protect and ensure the future of the land and the wildlife that we love. So my research is just a continuation of that work, and hopefully to steer us towards learning more about how we can more efficiently and successfully restore wild turkey when their populations to the areas that they are native to, when their populations are declining, and identify what populations are in need of conservation attention. So really, I'm like, that is the main goal of my work, and hopefully that will be able to steer us on the right path towards a secure future for wild turkey in Texas.
Yeah. Well, I really hope so. I really hope to see that happen because
Yeah.
I'm a big fan of turkeys, animals, saving the planet for sure. So
Mhm.
I really hope so.
Yeah. And so I think my work is, I'm like, I think I hope every scientist is approaching that their work is like, that with some aspect of like humility of that, like my work is a part of um, a larger effort. And so, I mean, it's not possible without all the people
that is true,
that are passionate and are all working together. So, my research contributes to that, but it is not all of it. Right. Yeah.
Okay, awesome. Okay. Thank you for answering all those questions. We're gonna shift a little more towards you, getting to know you a little better as a person.
Sure.
And see, how it all reflects back to your research at some point.
Mm-hmm.
So first question is, what do you do to motivate yourself every day, like to wake up and be like, let me go keep going with my research, stuff like that.
Yeah, I think a big motivator for me, because I mean, burnout is a real thing, imposter syndrome is a real thing, especially in graduate school, where it seems that everyone is expecting you to have all the answers and to know what it is that you're doing 100% and to like, to be completely competent and confident. So I mean, those are real challenges, and so I do struggle with burnout, and getting discouraged. So I think remembering why I'm doing what I'm doing, and remembering how important it is to me, because it's really easy to get short-sighted with a research problem because you have one question, right, like one research question, and it can be easy to forget beyond why that question matters, right? So trying to remind myself why it matters, is helpful for motivating me. I also rely a lot on the people around me. I have an incredible support system of friends, and family, and my husband, who support me in everything I do and continue to cheer me on and to surround me when things are hard, or when I'm running on very little sleep. They'll be patient with me and they'll encourage me. And so I have a lot of people that I love that, continue to push me forward, because I cannot just rely on my own strength. I need the strength of others. Did a beetle just fly into my head?
It flew by, but
it flew by. Okay, cool. I was like, there. But yeah, relying on my support system, helps to motivate me.
Yeah. Oh, awesome. So, what is something that you do or enjoy doing, especially when you're not doing your research? So, like anything you do for fun?
Yeah, there's a lot of things I like to do for fun. Now, whether I have time to do them, is another question. Yeah. But in my free time, I really like spending time with my friends. And that can be doing anything. I like going on walks or hikes. And I like to read a lot. And I like to read. And I also run a side business that's kind of on a hiatus right now, just because of how busy I've been. But I really like to sew. They're really constructive. But yeah, I like to sew. I like to make clothes and tailor clothes and make things for my friends and stuff like that. So, just depends on the season of life I'm in, how much I can actually devote to hobbies and free time. But I do, even in grad school, still try to prioritize like social my social life, and making sure I can see people, or else, I mean, I'm an extrovert, so I need the social interaction.
Yeah, that is awesome. Yeah. What's the most interesting part that you think, like whenever someone asks you, what do you do for your research? What's something that you're like, oh wow, this is gonna shock them? Yeah.
That I'm excited to tell them. I do come prepared with some pretty weird facts, that I will discover in the course of my research, so I can be really fun at parties.
Yeah.
So was the question, what are some things?
What is sort of like the most interesting part of your research that
I think I really like people are really interested and don't tend to know a lot about, like, you asked about the day-to-day and actually, so I think the actual process of working with genetic data, people
turkeys.
interested, and like what it's like to actually go into the field and chase around turkeys that were like driving around and trying to listen for their calls and trying to track them down, or the fact that you can. I just throw out some random facts, like the fact that you can tell the sex of an individual by the shape of its poop is like
Really?
like a really weird.
That's a real thing?
Yes.
Wow,
yes.
I did not know that.
Of a turkey, not anything else. Yes,
Okay.
turkey specifically, not just in general in life. But yeah, so that's something that people are typically shocked to hear. Was that more like the question you were asking?
I think that might not have answered the question, but that's an answer for sure. I love that though.
That's an answer.
Yeah, no. The real question is now, do turkeys fly?
Turkey. Okay, that's another thing people are shocked about.
See? Yeah, that's
turkey,
what I asked.
Yes. Okay. Yes. People have questions about turkeys. They can fly, not just like a little bit. They can fly well. Like I have come upon a turkey, and it has seen me and like looked at me, and then it has taken off and flown like above the tree line.
Really?
And like far away. Yeah. Wow. And I'm like, I can't even hope to catch up to you now. Like you're gone. You're totally gone. So turkeys fly. And that's part of their strategy for, escaping predators, right? Is they have these really large wings and they can take off really fast. So they're these big birds that any predator would just love to snack on, like a coyote, bobcat, an owl. But they can take off pretty quickly, and their flight is part of what helps them be so successful at evading predation.
Awesome, you wouldn't expect the turkeys to be flying. So whenever the animal does come by and try to snack on them, they just
they fly. Gone. They're gone. They're gone. And another fact. Okay, now I'm just talking about cool facts. Another fact that really does surprise people is that. So part of my work in the hill country of Texas was we were trying to actually trap birds so we could extract blood from them, so we could take some blood samples. So it was a team of me. I was the geneticist on the project, so a team of me and some other biologists, other grad students. We were sitting in a blind, and we set up a rocket net. So one way you can trap turkeys is by launching rockets attached to a net. Okay, and so people are always shocked to hear about that, and I was also shocked to hear about that. But you set up this huge net that's super long, and it's on the ground, and you try to cover it up with leaves and sticks, and then you set up these metal rockets that are attached to the net, and you wire charges inside the rockets, and you line like a fuse along the ground, and you are then sitting far away in like a blind, trying to be all camouflaged and concealed. And then you set out bait right in front of the net, and the turkeys are supposed to come to the bait, and you wait until all their heads are down. That's really important because you don't want to launch the rocket when their heads are up. You wait till all their heads are down, you launch the rockets, and it takes the net up and over them. And then it comes really down. Cool, huh? Yeah. And the net is huge. I mean, like, I don't even know, like the net is enormous, and it covers a big area. So when I tell you to illustrate the fact, like how fast these birds are at flying and making their getaway, by the time the net comes on the ground, some of the turkeys are already gone.
Really? They're that fast? Already
gone. Wow. And then they're all around the boundaries of the net. They're all around the periphery. There are no birds in the middle with the bait. They're all gone. They're all gone or almost gone.
Wow.
And that's we're firing rockets. So we're firing if that could just show you how quick they are at escaping. Turkeys are pretty dumb compared to most birds, but they are smarter than me. That's another fact people feel shocked about.
Okay.
Yeah. I mean, just by brain to body size, turkeys are one of the less intelligent birds.
But they are quick though. So
they're quick. So
that is awesome. Mm-hmm. By far one of the best responses I've had for those type of questions. Wow.
The more I think about it, I'm like, the more I was just like, and this thing, and this thing, and this thing.
Oh my God. I think that's it for the questions. I think that we're all awesome. Could we get a lab tour of where you work?
Yes, let's go.
Let's go.
Let's go.
Well, welcome to the GRACE Lab, stands for the Genetic Research and Applied Spatial Ecology Lab. This is where we store our field gear. So we have a lot of projects that integrate both field and lab work, as well as some that are primarily field work based. So a lot of our lab does bird research, so we'll have a lot of supplies for bird banding. We have binoculars, spotting scopes. In here, we have a lot of camera trap studies, or several camera trap studies. We have cameras, we have microscopes, we have bait, we have traps, all kinds of things for working with wildlife, trapping, collecting data in the field. And then this is our field gear section. So now we'll come to our proper lab section, and this line basically demarcates the field and the lab side. So now we're ready to enter the lab space. We've washed our hands, we've put our lab coats on. We will need to just put on a pair of gloves, and now we're truly ready. So this section of the lab is primarily for extracting the DNA from the samples that we collect in the field. So my project, I start with some kind of sample from the turkey. I start with either a feather, I start with a vial of blood, or a scat, and then this section of the lab, I work to isolate and take just the DNA from that sample to get this. And so the goal of the DNA extraction is to remove everything else from the sample, so the proteins, any contaminants, any inhibitors, any plant material that's a part of the sample from that material, so that all I'm left with is just the isolated DNA, and then I can actually conduct my analysis. So this is an extraction hood or a biosafety cabinet where we conduct all our extractions, and depending on the sample type we have, we don separate PPE for that. So if it's, depending on how hazardous it is, we might wear masks, goggles, just depending on the level of hazard and what kind of sample we're working with. We have various kinds of equipment to aid in our extraction or just any other kind of lab duties. These are some microcentrifuges. A lot of the equipment we have is we're working with very small quantities of material. So we have we have instruments that deal with really small tubes and really small quantities of DNA. Once you've extracted your DNA, we store a lot of our samples in these freezers, and long-term storage happens in the -80 degrees freezer, and that's really heavy-duty, very cold for long-term storage and preservation. So we have a lot of freezer space here, but then once we've extracted our DNA, the amount of DNA we actually have is not as much as we'd like or as much as we need in order to conduct our analysis. So basically, we need to take our extracted DNA to essentially a glorified copy machine. These are thermocyclers, all of these here, and they make lots of copies of our template DNA so that we have enough to actually work with. And these machines, these instruments, I'll call them, actually conduct what's called polymerase chain reaction, or PCR, and we add in an enzyme called Taq polymerase. We add in several like fluorescent dyes, and as well as primers that anneal to the DNA that bind to certain parts of the strand, the DNA strand, and then the Taq polymerase, through multiple cycles of heating and cooling, will actually make those copies of the DNA. We'll replicate the DNA strands so that we have a lot of copies to work with. So PCR is mainly done in this portion of the lab. We have the master cyclers, and then we have all of our tips and all of our pipettes that we need. We work with really small quantities. This is our nano drop. So after we've extracted the DNA, we want to know actually how well did the extraction work. We want to know the quantity of DNA that we extracted and the quality. So how pure is it? Were we able to effectively isolate the resulting DNA? And so that's what this spectrophotometer tells us. And it basically shoots light at the droplet that we place onto the instrument, and based on like the light that's reflected back and at which wavelength, it will then tell us the concentration of DNA that we've extracted. And so that's really cool. I really like that very low-key instrument a lot. I really like that instrument a lot. And this is a PCR hood.
So if we're working with samples that don't yield as much DNA, we will conduct all of our prep for the thermocyclers in this hood. And so this hood basically just prevents contamination from entering and degrading what little DNA we have. So a lot of our samples that don't yield as much DNA include feathers and scats because they've already been separated from the bird. So with blood, a lot of times it's much easier to preserve the DNA that's in there. You can put it in a buffer. You like basically take it from the animal itself and then you put it in some kind of preservation buffer. But a feather that's just on the ground, it's been exposed to the air, wind, rain, all kinds of animals, ants, and so the DNA is much more likely to be degraded. So that's why we have to be very careful when we're extracting those samples and amplifying that DNA so that we actually have enough to work with and that sample doesn't go to waste. So that's what the PCR hood is for. After you've amplified your DNA and you want to make sure that the PCR has actually worked, one way to test that is by conducting gel electrophoresis. And so you essentially load your PCR product into these gel chambers, and I hope you enjoy the names that we have for our gel chambers, thing 1 and thing 2. And this one's called the Grinch because he's very grumpy and doesn't play nicely with others. We load the PCR product into these gel chambers and we turn on these instruments here that run a current and moves the product across the medium, across the agarose gel. And then we take our mold into this gel dock, into this visualizer, and then we can actually see bands. So these bands will fluoresce because of the dye that we've added before we put it into the thermocycler. And so this is our ladder. And an analogy I've really liked to explain this is that the ladder shows you, picture like a classroom, okay? The ladder shows you all the kids. If all the kids showed up that day, this is what the classroom would look like. And then your actual bands, the actual PCR product is, these are the kids that did show up. So we want to see that some of the kids showed up and at this specific region. So at this specific location, this PCR was successful and we are seeing what we wanted to see. So that's really good. We also will use gel electrophoresis to test for the sex. So you can actually determine sex of an individual from its DNA. As you know, with humans, turkeys are the same way. A lot of organisms are the same way. So we'll use gels to, if it's male, sometimes depending on if it's male or female, you'll maybe see 1 or 2 bands, and then you can determine and score, okay, this one's male, this one's female. You can also use PCR and gel electrophoresis to test for the presence of certain parasites. So you can amplify DNA collected from blood samples, and then you can use markers that will anneal to the DNA of those parasites in the blood. And then if you have those parasites, you will see their DNA fluorescing. So we can detect for the presence of malaria. There are several, malaria is just an umbrella term for a group of parasites that behave in a similar fashion in the body. And so we can detect for the presence of different kinds of malarial parasites with gel electrophoresis. So that's a really neat and powerful way to assess the rate of disease spread throughout a population. So that's another aspect of turkey biology we're looking at with my project is with mine and some of the projects that are all, they're all intertwined, are looking at the presence of malaria in some of these populations and how much of a risk it is to these individuals. And yeah, well, I hope you enjoyed your tour. I had so much fun talking to you today about our lab, the work the Grace Lab is doing, and my research. Thank you so much for coming. I enjoyed talking to you about the work our lab is doing and about my own research. Thank you.